So this study is a study we actually started literally about 8 years ago when I had a fellow in the lab who was interested in nutrition. One of the things that we wanted to do is try and understand if individuals who had hepatitis C virus actually would respond differently to different dietary interventions. And it turns out that hepatitis C virus replicates on lipid droplets in the liver, and it steals some of the lipids from these lipid droplets to make its coat. And it was noted that saturated fatty acids in vitro system, in vitro system, the virus would bud better if there was saturated fatty acids around versus polyunsaturated fatty acids. And so we thought that perhaps if we actually gave a diet of polyunsaturated fatty acids to individuals, we could actually change the viral replication rate and perhaps make it more susceptible to dietary intervention or treatment in the long term. Now they have good drugs for hepatitis C, and they only cost $100,000. But when we designed the study, we actually thought about a different thing that we could do with the study. It's something I've been interested in. Is can we develop a unambiguous marker of dietary intake? So we wanted to be able to take plasma or serum and basically take a tube of blood from somebody and actually get an idea of what their macronutrient intake is and perhaps their micronutrient intake. And so we designed a study that we ran a lot of controls with. It turns out that people with hepatitis C aren't the most reliable subjects for long-term feeding studies, and so we actually abandoned that part of the study. One, it didn't look like it was working, and two, it was just too hard to do, unfortunately. So what we really wanted to do is, understand using these different platforms, can we actually get a predictive model of the system? And this system that we're looking at is dietary intake. So, so as you know, nutrients are inescapable, environmental substances. The effect of a dietary component is really dependent upon how much you eat, right? So if you eat lots of cholesterol, your cholesterol will go up a little bit. It's a lot of genetic determinants, but there really is differences, and we know that certain diets predispose you to the risk of certain different things. And so, so if you have folate deficiency or niacin deficiency, clearly you have a risk for birth defects and things like that. So our objective was to look at both short-term and long-term estimates of dietary intake. And so the study was, as I mentioned, we used an isocaloric diet in individuals. We estimated how many calories these individuals needed, and we prepared food. We gave them everything they ate for 6 weeks. And so it was 3 weeks on an isocaloric, high polyunsaturated fat diet where we had about 35 to 50% carbohydrates and 40 to 50% fat. And then we went directly to a second diet of high carbohydrate where we gave them 60 to 70% carbohydrates. We kept the protein content the same between both diets. One of the problems with dietary studies is that you have to change 3 different things, and then the subparts of lipids really makes it a little bit more difficult. And so we took blood from Metabolomics at days 2, 7, 21 of each diet, and then we looked at the phenotypic response based on this. And I won't talk a lot about that right now. And so this is the controlled individuals that we ran through the study. The gang in the Michigan Clinical Research Unit did a great job adjusting the diet, and there was less than about 0.75% body weight variation across the study in in the individuals. And so they did a really good job of keeping the diet such that they were not changing their caloric intake. And we did phenotypic responses, and I'll just show you the control subjects. We also ran a group, I forgot to tell you this, we ran a group of people with non-alcoholic fatty liver disease, not with hepatitis C, but with fatty liver disease that's associated with increased risk of diabetes, but these individuals were all non-diabetic. And so part of the phenotyping was just getting their lipid levels, and you can see that the high PUFA diet compared to the high carbohydrate diet had a bigger effect on a lot of the blood lipids. And we also looked at the glucose tolerance tests in these individuals. So we gave them 75-gram glucose or glucose load and looked at their blood glucoses at baseline and 120 minutes in. And what you can see is that there's a variability of responses among these people. So some people at baseline, their blood sugars will go down. Put them on a PUFA diet, they change a little bit. You put them on a high carbohydrate, they change a little bit differently. So there seems to be a phenotypic variation among these people, and this just shows two different patterns you can get. This person on the baseline, their blood glucose actually dipped below normal, dipped below their starting point at 120 minutes, and then when they went on a carbohydrate diet, actually went up, and this person actually did the opposite. So there's some phenotypic variations, and the question is, can you actually understand some of the basis of these phenotypic variations and what might be the best diet for somebody who's at risk for diabetes or some other metabolic disease? And then we did this with the non-alcoholic fatty liver disease individuals, and they were much more stereotypic. They had higher blood glucose no matter what diet they were on. The carbohydrate diet seemed to stop the rise as much. We looked at the liver function tests in these individuals, and you can see some people actually got better liver function tests on high polyunsaturated fat diet, and then their liver function tests went way up on the high carbohydrate diet, whereas one of the individuals actually went the opposite way. So we ran a bunch of targeted metabolomics on these individuals, and this is a heat map showing the relative relationship of at baseline. So these are the diets that these people came in on. We looked at the relationship, the correlation between demographic features and acylcarnitines, amino acids, their total lipids, and then we subfractionated the lipids into a phospholipid, diacylglycerol, free fatty acid, triglyceride, and cholesterol, so fractions. So we isolated those specific fractions in the blood, hydrolyzed all the lipids, and looked at the fatty acid chains that are associated with each one of those lipids. And then we just looked at how, across all these individuals, how all these things correlate with each other. And you can see the green line is the unity, and so so things that are highly correlated are green, things that are inversely correlated are red. And you can see that there is, on the baseline diet, there's a little bit of difference here, but you can actually start picking out some differences. If you look up here, there's sort of a modeled pattern in the acylcarnitine levels, where in the non-alcoholic fatty liver disease individuals, they seem to be less modeled, and I'm going to come back to this a little bit later. You can see there's a modeling pattern down here when you look at the free fatty acid fractions and things like that. And so we're really interested in how these patterns change, and I think Al is going to talk about this. I don't know if you're going to talk about correlation patterns when you talk. Okay, you're going to talk about it. So this is part of what Ala will go into, and she has a much more sophisticated way than we're doing it, but I'll show you some of the ways that we've actually utilized this. All right, so this is on baseline diet. After 21 days on the polyunsaturated fat diet, which you can see, and if you go back and forth, you can actually see in the controls, look at the screen here, and you go back and forth, and you start changing the relationships between all these different lipids, how they correlate with each other. And so if you can think about it, if you actually start increasing one thing a whole lot more, and everybody goes up on the same diet, that correlation is going to get better, especially between some of the lipids or the amino acids or the different things. And then you can use this kind of stuff to look at how the phenotypic characteristics that we have, the demographic sort of things, their body weight, which didn't change really, but their blood pressure, their glucose levels, and things like that. You can actually start looking at those correlations. Okay. Now, the most interesting part of this is when we put these people on a carbohydrate diet. Look at what happened to the non-alcoholic fatty liver disease group. Now, these people got a little bit less. If you look down here in the lipid pattern, you can see there's a little bit more monotypic sort of response in these different lipids. But you can see that the changes from a high PUFA diet to the high carbohydrate diet in the non-alcoholic fatty liver disease group was pretty robust. And this is really an interesting thing to think about how people who utilize high carbohydrates and carbohydrates when they're in excess in the liver, they're made into fatty acids. And so we believe that this represents a much more robust de novo lipogenesis in the liver of these individuals, and we're trying to investigate why that might be. So you can see then that this pattern is really sort of getting different, and these patterns are really important. And I'm going to come back to that this afternoon when I talk about, how you look at data. And so it's actually, we thought this was a pretty interesting sort of change in these individuals, and really led to a lot of other experiments. We did the same sort of analysis in a large group of people. I think it was about 30 people. We put on a weight loss diet from baseline to 22.5% weight loss after about 3 months. And you can actually see that the pattern here, and this is sort of that lower right pattern, but you can see there's a monotony pattern here before weight loss, and you get this much more modeled pattern. And this modeled pattern is looking at saturation, desaturation ratios in different lipids, because it goes 18-0, 18-1 fatty acids, 16-0, 16-1 fatty acids. And when you're overweight, or if you have non-alcoholic fatty liver disease, the ratios, everything goes up. But when you actually fast or lose weight, you actually decrease one versus the other, and you can actually start getting certain patterns. And it tells you that sometimes if you look at this kind of data, and I'll show this afternoon, is using ratios of different metabolites can actually become very informative when you start looking at data to try and pick out biomarkers of at least in dietary intake. It looks like it's going to help us look at biomarkers. So thinking about how you take this kind of data is, and I'll talk about this more this afternoon. All right, so when we looked at all of the targeted metabolites that we used, and we did a random forest analysis, what we found is that what was called the lipogenic index, which is the index of the 18-0 divided by the 16-0 fatty acids in the overall in the free fatty acid fraction, is the best predictor of what kind of diet you're on. So this is predicting the diet. And then the other lipid that was significantly associated with is this acylcarnitine with a one double bond. Now, this actually turns out to be a very interesting lipid. In the 18-1, with the intermediate in the oxidation of fatty acids, seems to be a good indicator of dietary intake. And so the question is why? Well, this turns out that these tend to be higher in individuals with who are on a carbohydrate diet. Okay, and what we believe is that when you eat a lot of carbohydrates, you do de novo lipogenesis. And high carbohydrate diets also activate the enzyme SCD1, which puts the double bond at the delta 9 position in fatty acids. And so you start accumulating those lipids in your blood. And so if you look here, triglyceride 16-0, actually it's this is also associated with, but there's 16-1 in here somewhere. What we think is that once these get into the circulation, they start getting metabolized by different tissues, probably mostly skeletal muscle, that when the trifunctional protein starts taking off 2 units, 2 units, 2 units, when it gets near the double bond, it sort of stutters. And that stuttering probably allows the CoA to actually get kicked out into the blood, and it's really a very sensitive indicator of how much lipid is actually going into the system. And so this looks like it may be an interesting biomarker to try and separate dietary intake. And we're working on this. Now, the other thing to see is that, there's ornithine is the only amino acid that made the list. The amino acids really didn't look like they associated with different dietary intakes. Okay. Now, when we looked at all the lipids, and so these are a subset of the individuals looking at their free fatty acid fraction at the individuals at baseline, then at 2, 7, and 21 days after being put on the PUFA diet or the carbohydrate diet, you can see that there's an abrupt switch, and you can actually start seeing changes in the lipid patterns in each one of these lipid subfractions. okay? And if you just simply do a statistical analysis and look at which lipids are changing, how quickly, you can see that some of these fractions have a very rapid change in their lipid pattern. So if you look at the triacylglycerols, after 2 days of feeding, you can actually see a statistically significant change in the 14-0 and the 16-0 fatty acid levels. And so you can see how some of these that change quickly might give you an idea of what the acute intake of food is, and some of them that change more slowly may be a much better indicator of long-term dietary intake. And we're going to come back to this a little bit later this afternoon, because now we've done lipidomic profiles with thousands of lipids and trying to understand these relationships. But from the targeted point of view, we can actually start seeing some very interesting patterns. And so this starts getting to where we're trying to go with the thing. What time is it? How much time do I have? Do we have a break after this? Okay. All right. So if we did principal component analysis of the lipid fractions, the predictors for the best predictors using principal component instead of the random forest comes up with some of the same things. Unsurprisingly, that the 18-2 predicts PUFA intake. Well, this is a polyunsaturated fatty acid that you only get from your diet, so that would be suggestive that you're eating a higher PUFA diet. 14-1, 61, and triglycerides in the phospholipid predicts dietary intake at 2, 7, and 21 days of carbohydrates. We've done some initial analysis looking at the relationship of different lipids and glucose levels and insulin sensitivity levels, and you can see that those individuals with the highest 16-1 free fatty acid in the blood, it's associated with a much lower glucose level and a higher insulin sensitivity level, but only when they're on the carbohydrate diet. When they're on the baseline diet, you can see there's a relationship, but you tend to lose it. But if you're on a high polyunsaturated fat diet, you lose these relationships. And so perhaps testing people under different dietary conditions might bring out some pathologies that you might not otherwise see. Interesting thing about the 16-1, it's thought to be an insulin sensitizer, so those individuals who are on a carbohydrate diet but maintain a high ability to make 16-1 look like they're more insulin sensitive, and this paper suggested that this may be a lipokine, although follow-up studies have not borne this out as well as one was hoping. Okay, so I probably will skip this. So what we want to do is find an unambiguous, reproducible biomarker of nutrient intake. Where we started this study is actually we took blood from these individuals and did transcriptomic profiling on the blood. And going through it fairly quickly, we could actually now start. This is sort of a harbinger of what we're doing in the lab now with the lipidomic studies. But what we found is that certain transcripts go up and down in peripheral blood lymphocytes under different dietary conditions. So this is on the PUFA diet. This is sort of by PUFA. So this transcript changed at day 2, had the biggest change, and day 21 it stayed changing. So we could actually find transcripts that change very reproducibly under different dietary conditions in peripheral blood monocytes. And these are upregulated genes, these are downregulated genes. On PUFA day 2, this is another one sorted on PUFA day 21. You can see there's some very robust changes in a lot of genes. And then you put them on a carbohydrate diet and they go backwards, they go the other way. And so there's a very rapid up and down changes in transcript levels, just like in some of the lipids that we see. And so the question is, if we actually take all this data and put it together, and this is just sorting on carbohydrate at day 21, so the biggest changes at day 21, you can see how these transcripts react under the PUFA diet. So some go up, but there's still some noise here as far as which ones are the ones that are responsive to carbohydrates. Okay. Interestingly, most of the genes that you see are up in carbohydrate feeding. So these are genes up and genes down, statistically significant changes. And so there's really a dynamic where carbohydrates seem to have a much bigger change in peripheral blood. And these are the pathways that are occupied by these differentially expressed, and oxidative phosphorylation seems to be the number one genetic pathway or molecular pathway that's changed. So we, without going into it, because this is what we're playing with in the lab, is actually looking at finding those genes that change the most robustly under different dietary conditions, both up and down. Extract those subsets of genes, and then rank order them under a lipid diet condition or a carbohydrate diet condition, and recognize how the absolute levels of these genes change in relative relationship to one another under different dietary conditions, because these are the ones that go up and down very robustly. And so if you're in carbohydrates and these go up and they go down in lipids, but the other ones go up and down, you can actually find the most responsive genes and order them. And then if you look at the correlation coefficient between the relative order of these genes, you can use a fancy way of doing a Pearson's correlation coefficient, and you can get these. You can get a pattern. And so then you take your blank sample, your sample, and you see how your blood orders, and then ask, can you make a prediction about what they're eating under these conditions? And so we got some blood, some data from a collaborator, UCLA, Michael Gorin and Chris Roberts, and they put some kids on a control diet or a low carbohydrate diet for 30 days. These were kids in a camp, and they drew their blood, and they did transcriptomics, so we got the data from them. And we took out the 100 genes that we had identified as the most responsive both up and down, and then used those to cluster individuals. And here's the control group that didn't get the low carbohydrate diet. You can see the baselines are interspersed with the befores and afters. Here at baseline, you can see that the low carbohydrate diet actually shifted everybody clustered over together using these specific genes. So it looks like you can actually do this. Now we want to do this with metabolomics. And so if we actually look at the correlation coefficient between these different ones at baseline, control, or low carbohydrate diet, you can see that there's a much higher correlation in those people who are on the low carbohydrate diet. So we think that by looking at the dynamic patterns of changes, you might be able to do this. And the question is, can you use this for other disease states? And this is what Al is going to talk about this afternoon, or tomorrow? Tomorrow. How you can actually now use the relationships between all these metabolites to really see the perturbations in the systems that you're looking at. And so we're really sort of excited about how this might work. So I'll stop there, and I'll allude to this a little bit this afternoon when we start talking about how to look deeper at these at targeted data. Okay? Is there any questions? I know I went through this pretty quick, but.
Is like a custom array, what you have?
Actually,
like
well, so these were Affy chips. Well, actually, these were Illumina platform chips. Yeah, the thought is you're exactly right. If you could actually set up your own custom chip, could you actually just use that to define a diet? We'd rather just do the metabolomics because it's probably easier. It's cheaper, at least, to do it that way. But we want to see how well these things correlate. And then I didn't have time. It's in your packet, but it looks like the genes that are regulated in different tissues are regulated similarly in peripheral blood lymphocytes. Can you actually start now using that to understand how different individuals, respond to different nutrients, and then go back to this whole phenotypic relationship? So, doing this multiscalar integration is where we're trying to go with it. Kawa.
Great talk. So if you look at the lymphocyte transcript, you see that good correlation with those metabolite change, or do you think?
Yeah. So, yeah, indeed we do.
Okay.
Yeah, so that's exactly what we want to do. So what are those transcripts that correlate with the changes in different metabolites? And how do they have some sort of functional relationship, or are they just, true and not functionally related? And which controls which? What we do is we do see an increase in SCD1, for instance, in the lymphocytes of the individuals after a high carbohydrate diet, which you predict, or people have shown actually happens in human liver after you eat a high carbohydrate diet. So we may be able to get some insights in the cause-effect relationship of different nutrients with different biological transcriptomic responses.
So because of accessibility, lymphocyte might be good, but
It turns out people don't mind that as much as they mind getting a liver biopsy.
Yeah. So, but if you have the choice of biopsy, so you think that getting liver is the best choice to see that effect?
Yeah. And so we actually have liver samples, but we're waiting on trying to do exactly what was pointed out. What are these genes? What are we really going to look at? The problem is getting controlled liver biopsies, because a lot of people get liver biopsies for fatty liver disease or suspected pathology. Sometimes we can get normal tissue from people who are undergoing the wedge resections of a cyst or things like that. But it's really hard under those conditions that are under anesthesia, and it's changing their metabolome and all that kind of stuff. So but then getting peripheral blood lymphocytes at the same time as you get the liver biopsy and look at the correlation between gene expression profiles across individuals. That's where we'd love to go, but money and time.
Is the expression profile different between non-obese versus obese?
They're different between the people with non-alcoholic fatty liver disease. The people with non-alcoholic fatty liver disease, we actually took those as lean, a BMI less than 27. So we don't really have, we have samples in the freezer from our weight loss study, just haven't gotten around to doing them. Or haven't found the money to do them. That's more the rate limiting factor. Anybody else? Okay, so I think we have a break now until what time, Terry?
Probably say 11.
Okay, so 11.